Thank you very much for giving us the opportunity to present our data. All right, so my name is Wataru Akahata. I am the CEO of the VLP Therapeutics. And in this company, we develop the vaccine against infectious disease and cancer. And I used to work for the NIH Vaccine Research Center. So today, as an introduction, I'm going to speak about my work at NIH. And then next, I'm going to talk about our malaria vaccine in my companies. So as an introduction, this work was done at the NIH Vaccine Research Center. Some people know this virus, but most people may not know this virus. It's called the chikungunya virus, belongs to the alphavirus. 2005 and 2006 outbreak happened at first in Africa, and then it goes to the Indian Ocean and India, and then Southeast Asia. Millions of people were infected, and it caused arthritis. And there is no drug, there is no vaccine yet. So NIH want to develop the vaccine against this virus. And as I said, chikungunya belongs to the alphavirus, and the alphavirus has this genome. So the alphavirus consists of the non-structure gene, 4 non-structure gene, and the structure gene. Structure gene consists of the capsid and the E1 and E2 envelopes. So E1, E2 envelope makes the dimer, and this E1, E2 makes the trimers. So luckily, we found that we insert the structure gene onto the mammalian expression vector. And the one strain express the VLP very well. And then this is EM picture, and then this is cryo-EM pictures. And there is no genomic RNA. That means there is no replication. Means it's safe, and the immunogenicity is very high, which immunize with this virus, the VLP, in mice and monkeys. And it shows very high neutralizing antibody because of the appearance is resembled to the wild type virus. So we published the paper, and we prepared a clinical trial at NIH. So of course it takes a long time, but finally the 30 people were immunized in a volunteer, and then it shows a very high immunogenicity. Actually, the much higher than the live attenuated vaccine. Live attenuated vaccine was developed a long time ago, but the side effect shows slightly the side effect prevents the more development. So compared to the live attenuated vaccine, the VLP vaccine induces much high neutralizing titer. And it's safe because it's me, and I'm still alive. So then I started my company and tried to explore more vaccine development. I began to work on platform technology against many virus or many cancer. We want to make the cancer vaccine and the infectious disease vaccine. So it's called the plug and play platform. Inserted alphavirus virus particle vector, so we call it i-αVLP, and the target antigen are displayed on the surface of the VLP. And the high titer of the antibodies against the target is induced. So how we are going to make it? It's very simple, but based on the structure-based design, we at first we make the more stable VLP, and foreign antigen up to 50 kilodalton can be inserted into 2 different sites of the envelope domain. So E1 and E2 has a heterodimer. That's 240 copies of the E1, E2 envelope is displayed on the one particle. So actually, 480 copies of the foreign antigen can be displayed on this platform technology. So one of the advantage of using the alphavirus VLP as a platform technology is this. Antibody has two arms, and then this antibody has a bivalent binding. So, and then usually the typical IgG has a wingspan is 10 to 20 nanometers, and then they recognize exactly the same antigen. So when you insert foreign antigen onto the alphavirus, actually the distance is 11 nanometer. It's kind of the ideal distance to induce the ability of the antibodies. The B cell stimulate very well using this system. So the summary of the advantage of the i-αVLP technology is compared to with other VLP product is superior antibody induction, symmetrical and repeatedly display of the antigen, ability to display 480 copies of a single antigen, or the 240 copies of the two different antigen per VLP. And the larger particle, this is a 70 nanometer particle. So the larger particle accommodates larger antigen up to 50 kilodalton. And now I'm going to talk about the malaria i-αVLP vaccine. It's a VLP M01 development. To prove of this platform technology, we start to try to make the malaria vaccine because of obviously the half of the world people has a risk to infected the malaria infection. So one of the most difficult, challenging things to make a malaria vaccine is that malaria has a very complicated life cycle. So that they have a 3 stage of the life cycle. So sporozoid form, and then blood stage form, and then transmit form. So once the malaria has infected into the body, it is very, very difficult to eliminate the malaria outside of the body. So but the long history of the malaria vaccine shows some hint to make the vaccine. So it's called a CSP, is proved to be effective, and the CSP is in the sporozoid home. The CSP is a surface of the sporozoid of the protein. And then monoclonal antibody against CSP, a repeat domain, prevent malaria infection in preclinical model. So the CSP has a repeated motif of the NANP motif in the central domain. So the monoclonal antibody recognize that domain it prevents the malaria infection in a preclinical model. And also it is suggested that the protective threshold for anti-CSP IgG concentration in plasma is more than 20 microgram per ml is probably enough to prevent the malaria infection. So from those data, we insert the CSP NANP repeat sequence onto our VLP. And this is EM picture, and then this is cryo-EM picture. So the cryo-EM picture was provided by Michael Roseman's group in Purdue University. So as you see, the particle is very stable, and then the CSP epitope is exposed on 400 copies of the particle per particle. And we immunize with the mice. So we use the arm adjuvant because of the arm adjuvant is the cheapest, and then everybody can afford to buy. So we choose it. And then I know that as an adjuvant can enhance more antibodies, but to make the malaria vaccine development, we want to stick to the arm adjuvant. And as you see, the 0.5 microgram and 1.5 microgram, 50 microgram induce very nice antibodies against the CSP protein. And this is we immunize in a monkey with the VLP M01. And this is a comparison. It's not a direct comparison data. I took from the paper. So one of the leading candidates is called RTSS, developed by GSK. They are using also the VLP, the vaccine, using the not exactly same, but the targeting to the CSP. And they are using adjuvant, nice adjuvant, AS01. And they immunize in monkey, they induce the antibody titer around 3,000. And then after second immunization, they show around 10,000 antibody response. It's called the OD 1.0 titer. And we also immunize in monkey, and the first immunization is more than 10 times higher, and then after the second time, more than 10 times higher induction. And then this RTSS shows around 30 to 50% protection rate in phase 3 clinical trial. So one of the reasons why compared to the RTSS, we have a much high antibody is that we insert the 100% of the alpha-VLP. It's much bigger, larger the particle. They are using HBV-VLP platform technology. So that because of the small of the particle, they cannot insert 100%. So that they end up 25% of the CSP HBV fusion protein mixed with 75% HBV-VLP. So therefore, antibody response is not quite high as compared to ours. That's my the hypothesis. So then just proof of our antibody is effective or not. So we did the passive transfer of the serum immunized with VLP M01 in monkey, and then challenge in mice model. So we collaborate with Dr. Fedel at Johns Hopkins University. So he has a chimeric mice malaria model. So his malaria expressed CSP, human CSP, but the other one is mice malaria. So that it infects the mice, and then we can look at the protection rate, and then obviously the ones after the monkey passive transfer, after the serum, after the passive transfer from the serum in a monkey immunized with our VLP M01, completely protected against his chimeric malaria challenge. And as one more other proof of the concept, we did the mice malaria vaccine development. So we completely changed the CSP epitope from the human malaria to the mice malaria epitope. And we immunized twice, and then we challenged to the mice malaria sporozoid. And as you see, the control is infected 9 out of 10, and our vaccine protected the 9 out of the 10 the mice. So I think we believe that this vaccine can protect the malaria the infection much more than higher than the GSK vaccine. So actually, our target is only the travelers and the military, because of we cannot distribute to the whole the world. Just at the first target is the travelers and the military. But right now, this is our clinical development stage. So the pre-IND is done, and then probably we are going to do phase 1-2A clinical trial in 2016 at the Walter Reed, and then we are going to move forward. That's our plan. And the collaboration with, as I mentioned, Johns Hopkins University people and the Purdue people, and then we have the nice scientific advisor, the Stan, and we are going to move forward. So this is my last slide, acknowledgement slide, and I always get advice from the co-founder. They made their own company, and then they have successful. So that I got advice every time when we have when the clinical development is sometimes it's so complicated. So those people are helping us, and the collaboration people and the scientific advisor. Thank you very much for paying attention.